Right, today we're going to be learning about mutations and repair. So so here's like a table of contents. We're going to go through characteristics of the genetic code, so like DNA and RNA. We're going to go through all these different aspects of mutations, uh, and then we'll get to proofreading and repair, and we won't do transposons. So there are a couple of characteristics of the code. First of all, the genetic code is universal, so all living things use the same 4 bases. And I guess for RNA, uracil is a fifth base that's included. And all living things use the same code. So this chart, the way to read it is first of all, over here, refer to the first base of your codon. And then if it's like first row, then you look at the columns, which is the second base in your codon. So let's say UC. And then for the last base, you basically go on the row in this little box. So if I want to do UCU, I go U over here, C, and then I look at U. And this is basically, remember yesterday David asked if there are 4 times 4 times 4, 64 combinations to arrange 4 bases, but only 20 amino acids, then how does that happen? This is how it happens. Yeah. So all living things use the same code. This codon chart is applicable to all living things.
Wait, so just like a semantics thing, why do they use like do you know is there a reason why they use U instead of T?
Because on the RNA, which is what codes for the codons, uracil is
Oh, okay.
replacing thymine. Yeah,
Okay,
so
so you
that's
go
why
by
they use U.
RNA. Okay.
Yep. Right, so Nierenberg in 1961, he deciphered the first codon. He got a Nobel Prize for this, and for his interpretation of the genetic code, which means he found also other codons. So what did he do? He synthesized an artificial mRNA with identical RNA nucleotides, something like this, and therefore there's only one type of codon in here, and he found that this mRNA only resulted in one type of amino acid, or in general, if you only have one type of codon, then it only results in one type of amino acid in the polypeptide. So he also repeated this with other nucleotides, and he used other techniques to decode mixed triplets. So if this is just a UUU, then if there's like a UCU, then that's a mixed triplet. So there are 64 different possible codons, but only 20 different amino acids, which creates a redundancy. So what that means is if you look at proline, more than one codon here can code for proline. So essentially what redundancy means is you have like a failsafe. So if your CCU is, let's say potentially like in terms of what a failsafe means, it's if you have a CCU like not working, then you still have a CCC, a CCA, and a CCG to still code for proline. And vice versa, like if CCC isn't working, then you have a CCU, CCA, and CCG. That's what redundancy means. And at the same time, there's also no ambiguity. So if you have a codon CCU, you will know that no matter what, it will always code for proline. CCU can only code for one amino acid, and all the other codons, they only code for one amino acid, although a single amino acid can have multiple codons for it. Alright, so there are 64 different codons on mRNA, which you already knew, 20 amino acids, which we already learned, but there are also only 45 different tRNA molecules. So what does that mean? If basically the way to ask it is, or the way to think about this is here. So if we know that tRNAs need to have like a 3 base anticodon, then shouldn't there be 64 different tRNA molecules corresponding to the 64 different codons on mRNA? Well, the answer to that is that some tRNAs can actually recognize more than one codon. And that's where we learn something called the wobble hypothesis. So, the wobble position is the third base in a codon. So whenever I say wobble position, that's what it's referring to. And there's a definition here. The third base of the mRNA codon and its corresponding tRNA anticodon. That is the wobble position. And in this wobble position, base pairing rules are a little bit more flexible. So let's take a look over here. Let's go back to the structure of tRNA and the anticodon. So in the third, I don't know which way it reads. Wait, 5 prime to 3 prime, right? So I guess the third one must be here. But moving on. In the third position, no, it's anti-parallel, so it would read 3 prime to 5 prime because mRNA is going 5 prime to 3 prime. So yeah, in the third position, the rules are a little bit more flexible, and that's because there's another base apart from U, A, C, and G called inosine. And inosine is a modified form of adenine that's found in tRNA's anticodon. And inosine can form hydrogen bonds with U, C, or A on the mRNA, which allows for a little bit of flexibility. So if we look over here, for example, tRNA, let's say that it has an anticodon of CGI. And what that means is if you have CG, oh, that's a terrible G. If you have CGI, then if this is on your tRNA, oh, I'm running with them. That's pretty bad. Hopefully you can understand. So if you have a tRNA here, and then you have your mRNA here, I won't write the entire thing, takes too long, then potentially what can this tRNA bind to? Well, G and C are not flexible, so this has to stay. But earlier we said that inosine can bind to U, C, or A. Then this can be a GCU, it can be a GCA, and it can also be a GCC. And you can see that here. GCU, GCC, GCA. And all three of these codons can be paired to by a single tRNA molecule. And what does that result in? So if a tRNA, a single one, can bind to all of these, then essentially what that means is that this single tRNA carrying an amino acid, let's say it's alanine, so that's why GCU, GCC, and GCA in this particular example can all code for alanine. So this is one way to, this is a hypothesis. So it's a pretty good hypothesis if you ask me. I don't know why they still call it a hypothesis if it hasn't been proven or not. But yeah, maybe we could ask Miss Law that question if she sees the video. Alright, so now we'll cover mutations. A mutation is a change in the genetic material of an organism. Uh, and a genetic disorder or a hereditary disease is a harmful mutation. So these are harmful mutations in gametes that are passed on to the next generation. And gametes are like sperm and eggs. So there can be several origins or causes of mutations. Mutations can be spontaneous, so it can be like an error in genetic machinery. So DNAP can make a mistake, or yeah. Or it can be due to other enzymes. They can also be induced, so mutations can arise from exposure to certain mutagenic agents. And there are also things called transposable elements that can cause mutations. So errors during crossing over can create mutations, and something called transposons, which we won't cover today, can also cause mutations. So, what is a mutagen? A mutagen is a substance that can cause genetic mutations. So they are physical and chemical mutagens. physical mutagens can be radiation, UV light, or X-rays. Which, yeah, okay. UV light and X-rays they're both just like light, different frequency, and then radiation is like alpha, beta, gamma particles. Chemical mutagens can be base analogs, intercalating agents, or base modifying agents, and these are all different chemicals, and we'll see them in a moment. So let's start with some physical mutagens. UV light. So this is a very common cause of DNA damage, and UV light can produce something called pyrimidine dimers, or a type of fused base. So if you take a look at this example, you have two thymines. They're both pyrimidines, or here T and C, which are both pyrimidines in this example. What can happen between them is with UV light, there can like you can basically like create two covalent bonds between these two pyrimidines, and they bond to each other, creating this. And what happens in the actual DNA molecule is when these two pyrimidines bind, it kind of creates like a kink or a bend in DNA. Yeah. Uh, this is an example of a disease caused by dimers or pyrimidine dimers, but it's beyond the fact that you have dimers. This disease is characterized by the inability to repair damage caused by UV light. So a mutation in an enzyme in the here NER. I think we'll learn what this term is in just a second. Individuals, yeah. So basically, remember we said that UV light creates pyrimidine dimers. So that's why people with xeroderma pigmentosum may need to avoid sunlight completely because UV light from the sun can create these dimers that they do not have the ability to correct. Let's look at an example of a chemical mutagen. Base analogs. So base analogs are structurally similar to normal DNA bases, so they can almost mimic them, and they can get incorporated into the DNA instead of a normal base. So for example, thymine can be replaced with 5-bromouracil. So what happens? Let's look at this example. Base analogs can pair incorrectly or pair incorrectly during DNA replication. So for example, 5-bromouracil can pair with G instead of A. So if thymine normally pairs with adenine, 5-bromouracil will pair with guanine instead. So what happens here is, let's take a look at like normal replication. So on this side, the bottom strand has been taken as the parent. So when the bottom strand is taken as the parent, you're perfectly fine. Or this side is perfectly fine because adenine will bind or will pair with thymine, which is normal, and then further down the line, you won't notice because you have normal adenine and thymine. But on the other side, when you take the top strand as the parent, what happens is 5-bromouracil is pairing with guanine. And if you go one more replication cycle, this guanine will then pair with a cytosine. So what you have now is a base pair substitution. This base pair is completely changed. And the other side, once you take bromouracil again, it's going to pair with guanine another time, and essentially what happens here is going to keep happening. So interestingly enough, this is used in the drug for treating HIV. Let's take a look at another chemical mutagen, something called an intercalating agent. So these guys distort DNA helices by intercalating between adjacent base pairs. So they basically go in between nitrogenous bases. And what can happen is intercalating agents can cause DNAP to stutter and copy the mutagen as an extra base pair, which is bad, or can be really bad. They can also interfere with replication, but they're commonly used for staining and visualizing DNA in biotechnology. And the final chemical mutagen, something called a base modifying agent. What happens is these base modifying agents, they modify bases, changing their pairing properties. So in this example down here, this adenine base has been modified and altered so that instead of pairing with thymine, it's going to pair with cytosine. And let's see what happens. When you take the top strand as the parent, you create normal daughter DNA. But when you take the bottom strand as the parent strand, adenine, which is normally supposed to pair with thymine, is going to pair with cytosine, and that's going to create mutated granddaughter DNA in further replication cycles. In this example over here, you have a base pair substitution again, and over here you still have this altered adenine base, which is going to create more mutated granddaughter DNAs down the line. So let's look at a little overview of types of DNA mutations. You can have chromosomal mutations, which can occur during meiosis or crossing over. You can have missing bases, fused bases, like the dimers we saw before, or mismatch mutations. They are incorrectly paired bases. They can be caused by point mutations, and they're one of the most common types of errors during replication. Cool. Let's look at some different types of point mutations. And a point mutation is when one nucleotide or a single base pair is altered. So first of all, let's look at substitutions. Substitutions are a small change in a DNA base pair where one nucleotide is replaced with another. So in this example, the cytosine has been replaced with uracil. If you look here, there's something called a transition or a transversion. A transition is when a purine is replaced with another purine, or a pyrimidine is replaced with another pyrimidine. And a transversion is when a purine is replaced with a pyrimidine and a pyrimidine with a purine. So yeah. Now let's look at frameshift mutations. Yeah. So frameshift mutations, their name comes from basically the effect that they can have on DNA. So normally there's something called a reading frame, or not normally, but there's something called a reading frame. And a reading frame is the triplet grouping of a genetic message. So remember in the last lesson where we had like the words and you had this basically at the front you had like a block of like incomprehensible letters, then you had the to start the message, a sentence with three letter groups in between, and then the message would end with end and then with like more incomprehensible stuff afterwards. So what the reading frame is the triplets or the three grouped letters that we could read the sentence by. When your reading frame is moved, it's essentially like moving your reading frame. Instead of starting with the, you're going with H, E, and whatever letter afterwards, and then you're shifting your reading frame by one letter like this. Really confusing for us to read, and pretty devastating to like genetic material like proteins that are going to be created afterwards. So, what is a frameshift mutation? A frameshift mutation is when the number of nucleotides that are added or lost, so it's not being replaced in this case, added or lost is not a multiple of 3 and you're altering the reading frame. And this can happen by an insertion or a deletion. An insertion is an addition of one or more nucleotide pairs in a gene, but a deletion is a loss of one or more nucleotide pairs. So you can also have no frameshift if a number of nucleotides added or lost is a multiple of 3. Because if you move the reading frame exactly by 3, then you're still reading the three letter groups that you're supposed to be reading per se. And this can lead to an extra or a missing amino acid. Let's look at some diagrams. So let's look at a frame shift deletion. So normally in this wild type, which is the normal per se, a uracil has gone missing. So normally you would read this UUU and then the GGC and a UAA, but now without that U, once that U goes missing, you have an AUG, AAG, now you have a UUG, a GCU, And normally where you had this stop, there's no stop anymore, depending on, what comes later. So this stop, which is normally supposed to be here, you're no longer reading it. Let's look at this, a frame shift insertion. So you start with an AUG, and now with this extra U, you immediately have a stop. So this is a frame shift insertion. The thing to focus on here is the fact that this extra U is changing your reading frame. And let's look at an example with no frame shift mutation. So the reading frame is preserved because you start with AUG, you lose 3, so now your reading frame is still preserved.
Well, yeah, I have a question about let's say what if instead of AAG being deleted, it was like GAA, for example. Would that still be a frame shift mutation?
GAA,
Mhm.
that is a good point. Oh, that is a good point. That's a good question.
Because it would I guess it would mess up the reading frame in some way.
Um, Ask Miss Law that question. I don't know if I have an answer for you. That's a very good question.
Okay.
Yeah, for sure ask her that one. Alright. Yeah, that's a very good question. I'm curious about that too. Because one way you could say that, I mean, in this case, you would still have the AUG to start, and in this case too, like you would still have the groupings of 3 afterwards,
They would be completely different amino acids.
but it would be completely different amino acids. That's a good point. So right now we're just looking, yeah, that's a good question. So we know the different types of mutations, but now we also classify the effects that these mutations can have on an amino acid sequence. And with these effects, the general effect on protein function itself. So let's look at this. A missense mutation is when you code for a different amino acid, or the effect of a missense mutation on an amino acid sequence is a different amino acid. A nonsense mutation is when you code for a stop codon, which isn't supposed to be there, and the effect is that it results in a truncated polypeptide. And a silent mutation is when you code for the same amino acid, because remember the redundancy? So there's 3 different classes of mutations, but they're caused by all the things that we were looking at before. With a missense mutation, protein function may or may not change. It's hard to tell. So for an example, sickle cell anemia, sorry, when you have a single point mutation, and you have an A instead of a G, the protein function does change, but it's not catastrophic per se, because if you have sickle cell anemia, you can still live with it, right? Let's look at a nonsense mutation per se, because this is what you have to compare it to. So you have a stop codon which isn't supposed to be there, and you have a truncated polypeptide. Most of these proteins are not functional at all, right? They're not. So in this example, you're supposed to have 4 amino acids in a stop, but now you have methionine in a stop. So this protein is not functional, and most of these proteins are just digested by the cell. And mutations or nonsense mutations at the embryonic stage are often lethal. And a silent mutation, which is a mutation that basically you end up with the same amino acid just with a different code. This does not change protein function at all. So a missense mutation is when one one amino acid has changed. A nonsense mutation is when you have a unexpected stop codon, and a silent mutation is when you have a different code, but it codes for the same amino acid anyway. If that makes sense. Any questions? And remember that a frameshift mutation can cause extensive missense, so many different amino acids have changed, or even a nonsense if a unexpected stop comes down further down, right? So let's look at sickle cell anemia as an example. In this, it's a substitution missense mutation. Adenine is changed to thymine, and this changes the amino acid glutamine to valine. These are sickle cells. These are normal red blood cells. Let's look at a nonsense mutation, like we said before. So this is the normal amino acid chain, or sorry, normal polypeptide. In this case, you have a U instead of an A here. So this is a point mutation. Or, sorry, is this a point substitution? Am I using the wrong term? Point substitution. You have a U instead of an A. So normally where you had an AAG, you now have a UAG, which creates a nonsense. You can also have this insertion with an extra U, causing immediate nonsense. Or a frame shift causing an immediate nonsense. Yeah. Or a silent mutation where U instead of a C, GGU, GGC, both code for glycine. So overall, no change. So here are some examples. So, two men sat and had hot tea. This is your wild type. So this is what your sentence is supposed to look like. Example 1. Two men sat and had hot tea, capital S. Uh, Lowercase s, capital S. Does the information in the sentence change?
I don't
Anybody?
think so.
No.
No.
Yeah. So it's still two men sat and had hot tea. Silent. Or I guess first one, silent. Second one. Two men sat and had hot C. Has the information changed?
Yes.
Yeah, so whereas you had T before and you have a C now. So if each three-letter word is one amino acid, your last amino acid has changed from T to C. So this would be considered a missense. Let's look at this. To me. Has the sentence changed?
Yeah.
Yeah. The sentence has unexpectedly ended after me. So you have a nonsense mutation here. Let's look at this. Two men sat and hot to ah. Uh, what we have here is a frame shift insertion. So this insertion here has changed my reading frame, or changed the reading frame. And over here, two mes ata and at yeah. Uh, yeah, another example of a frame shift deletion. So by n being deleted here, our frame shift, you have now have a frame shift mutation, and you have no clue what's going on here. Basically, all your amino acids have changed except for two. Alright, so remember, there's a difference between the types of mutations, so point mutation, substitution, frame shift, insertion, deletion, or no frame shifts, and the effects that they have on your polypeptide, and further down the line, the effects that will have on protein function. So just because you have missense doesn't mean that your protein will go from functional to like non-functional. It can have a negative effect, it can have a neutral effect, it can even have a positive effect. So here's a little summary. Point mutations and substitutions, they can cause missense effects. They can cause nonsense effects, but they can also just be silent. Frameshift insertions or deletions can cause extensive missense, nonsense, or just be silent. Or sorry, they can't be silent, which is what this X means. So a frameshift insertion or deletion can never just, code for the same amino acid because you've created, you've added in another base or you've taken out a base. No frameshift insertion or deletion. So an extra or missing amino acid, it can have a missense effect. It can also have a nonsense effect, and it can never be silent. All right. This is just another uh, overview slide. So if the effect on the organism or protein function is negative, then that's detrimental to the organism, and this can be caused by missense or nonsense mutations. Yeah, most molecular biological research is related to this idea. Positive mutations, they benefit the organism. For example, like back mutations can reverse or restore the original sequence or confer antibiotic resistance, or neutral or no changes. So mutations in non-coding regions, so they're silent or sometimes cause missense mutations. So now we get to proofreading and repair. So we've covered all these different ways that mutations can happen and the effects that they can have, but how do we avoid them? And that's by proofreading and repair. First, we'll talk about normal error rates and error rates after proofreading and repair, and we'll also talk about the different ways that our body repairs DNA. So the average human chromosome has 150 million base pairs. Initially, the pairing error is 1 in about 10,000, so about 15,000 errors per replication, right? But after proofreading and repair, you have about 1 error in 1 billion base pairs. So 0.15 errors per replication, or a single error every 7 replications. No, did not have to memorize these numbers. This is like a fun fact. Yeah. So, yeah. What happened in between the initial and final error rate? So what happened is proofreading and repair. Yeah, proofread and correct your essays, guys. Like if you're normal, let's say you have an essay, your normal error rate is about 15,000 errors per essay. After proofreading and correcting your essay, you might have 1 error in every 7 essays. Yeah, that's a lie, but good example. So, repair by nuclease. A nuclease is an enzyme that can break phosphodiester bonds in DNA, and they can excise out the nucleotide. An exonuclease binds to the end of a nucleotide chain, so they can cut off. So for example, an exonuclease, like let's say your DNAP, which is an example of an exonuclease, is going along, doing its thing. It's it can basically like cut off this last nucleotide and then like replace it, just to go back and correct its own mistake. An endonuclease can bind to the middle of a nucleotide chain. So let's say you have a DNA, it can bind to the middle of it and cut things out. So let's talk about what this does. An exonuclease proofreading. So this is considered instantaneous repair. So this occurs as the DNA is replicating. DNAP3 and DNAP1 both have exonuclease activity. So how does it do this? DNAP can instantly recognize mismatches during replication. So when it does recognize a mismatch, it will hydrolyze the phosphodiester bond, releasing the last nucleotide that was just added. This is the exonuclease activity, and then it can replace it with the correct nucleotide, and that's its polymerase activity. So note that the one enzyme, DNAP, does both the nuclease and polymerase function. Pretty cool. Now let's look at endonuclease proofreading. So this proofreading can repair or repair often occurs. So this repair often occurs after DNA has already replicated. So this mechanism of repair is known as nucleotide excision repair, and that's the NER we saw earlier at uh, over here. Yeah. So xeroderma pigmentosum. Yeah. Excuse the picture example if it disturbs you, but yeah, this is what is meant by NER, nucleotide excision repair. Let's go all the way back. Right. So how does NER work? An endonuclease can recognize and bind to errors in DNA. And what it does is it nicks the strand by breaking phosphodiester bonds before and after the error, and then it can excise this section of the DNA. And remember, this is just one strand though. It's not excising like both strands, right? It's just one strand over here, and it pulls it out. And then what happens is a polymerase will come by and replace the gap with the correct nucleotides, and then a ligase will come, and then it will seal the nick over here. This example can show you pretty well too. So this is a section of damaged DNA. An enzyme complex, or the endonuclease that we talked about, will recognize this distortion. It'll come here, separate the DNA, use single strand binding proteins to stabilize the single strands. It'll cleave the strand on both sides of the damage, and then part of the damaged strand will be removed, and then DNAP and ligase will come by and fill in the gap. Yeah. So let's look. I mean, we can look at some questions. The lesson today was pretty short. So the genetic code is redundant but not ambiguous. Do you guys know what that means? Any answers?
There's multiple like examples that can multiple codons that can pair for an amino acid,
Mm-hmm.
but there's only it only creates one amino acid. It's not ambiguous.
Exactly. So multiple codons can code for the same amino acid, which is redundancy, but one codon cannot code for more than one amino acid, which would be ambiguity. Yeah, it's good. So now in this diagram on the right, anybody know what's going on here? You don't have to, we won't go through each step, but just, yeah. So what's going on here is what we learned. It's good to know what's happening at each step. It is beneficial. So if you guys have the time, I would recommend going through each step. Alright, so we won't be covering transposons. Do you guys have any other questions? Because that's the, oh, what's going on between the last two steps? Okay, so I'll just go through step by step then. There's an error here. So at this point, an endonuclease would come and recognize it, nick the strand before and after, remove it, which is happening in step 3. Step 4, DNAP and DNAP would come, yeah, exactly. DNAP would come and replace the nucleotides that were excised, and then David suggested ligase maybe in the chat. Yes, David, that you're correct. So what would happen here is, assuming this is 5 prime and this is 3 prime, then we'll say that the nick is left here, and ligase in the 5th step will come and seal the strand over here. Anybody have more questions? Alright, yeah, if there aren't any more questions, that's the lesson for today. Pretty short. Uh, yeah.